Hello, Assalamu alaikum. In this video, we will understand about the chemotactic and phototactic movement in bacteria. So, the chemotactic and phototactic we will collectively understand. So, chemotactic mean the chemical, the chemokines can be, and the phototactic mean the light. So remember, the photons mean the light. So tactic mean the movement from one place to another. So in this way, here is the bacteria which that contain a MCP protein which that bind with the chemoattractants or the chemorepellent, and the bacteria which that will be moved through flagella activation. And in this way, for example, here is the skin cell and the gut epithelium and free living bacteria and eukaryotes which that release the cytokines or chemokines basically, which that recruit by the binding of the chemoattractant lead to reside the bacteria microbiome in our skin and our digestive system, for example. And the light, which that is also important. Why? Because the photosynthetic bacteria required it for the photosynthesis. So in this, the bacteria will move toward the light for the photosynthesis. The decrease the distance. In this way, the response of the chemoattractant will also decrease. The random movement and non-random movement will occur due to the distance and closed. For example, the distance will lead to finding behavior and activate more energy expanding. In this way, bacteria will find the location. But the non-random movement means whenever the exact movement due to closer and more chemokine to reduce the response and the saving energy by chemotaxis. And in this way, the exact location and the light basically also closer to reduce response. In this way, the bacteria will reserve the energy by this mechanism. For example, the decrease the distance toward the chemoattractants where the source of the releasing of the chemoattractant. In this, the bacteria will lose their ability to respond but automatically find the location. Very easy. So here is, you can see, this is the bacteria which that is contain a MCP protein as a receptor. So for example, the attractant and repellent which that is involved for the bacteria movement. For example, the repellent also activate the flagellar movement and the attractant also. But the negative phototactic movement means the away from the light, while the positive phototactic movement means the toward the light. In this way, the chemotactic movement are also the positive and negative. Here is the retinol or other type of the light absorbing protein. The orange light and the UV light is important for the absorption. In this, the HDR receptor which that receives the light and the MCP protein which that trigger in this way will lead to intracellular domain which that is contain a basically is the SR sensitive receptor activation will lead to Chi protein. Chi protein activate the methylation intracellular domain will lead to Chi-W, and the Chi-A complex formation will lead to phosphorylation, and in this way the Chi-B protein will, whenever the phosphorylation will lead to demethylation when the bacteria will closer toward the light, and in this way the inactivation will occur. The methylation activate and demethylation will lead to inactivate. In this is the Chi-Y protein activation by the Chi-W protein and the Chi-A protein, Chi-A mostly. Chi-Y protein phosphorylation will lead to activate the flagellar movement by the binding of the intracellular domain of the flagella. The motor of the flagella. So the flagellar motor will activate, lead to clockwise rotation and the counterclockwise rotation. The counterclockwise rotation unidirectional whenever the bacteria will closer and closer more. In this way, the cell movement will occur. In this way, the clockwise rotation means whenever the distance will be more, so the finding behavior will be more activate. In this, the consumption of the energy will be more, and that is why the more consumption of energy, in this way the chemotactic movement randomly more. So this is the reason, and let's understand about whenever the bacteria will closer to the light or the chemotactic closer in the chemoattractant. So for example, a light. So whenever the bacteria will closer and closer, so the detachment of the Chi-Y protein phosphorylation, and in this way the Chi-Y protein will be dephosphorylate by the Chi-Z protein, that complete mobility mission whenever. But anyhow, here is the attractant and repellent we will understand now. Then we will understand about how it is possible to decrease the movement and the Chi-Y inactivation. Here is the attractant and repellent. When bind with the MCP protein, will lead to methylation activation due to the Chi-R protein. So the Chi-R protein same for the photoreceptor, the phototactic movement, and the Chi-R protein activate the methylation, and this will lead to methylation to Chi-A protein activation, the phosphorylation due to the Chi-W protein. The Chi-W protein is basically activate the Chi-A protein by the internal, basically is the cytoplasmic domain. Activate Chi-A protein increase the Chi-A activity. So the phosphorylation due to the less chemoattractant or chemorepellent, and in this way the less chemoattractant and less basically chemoattractant will lead to increase the Chi-A protein activity because the finding behavior. So the bacteria will quickly find the location of the source of the chemoattractant. So finding behavior will be activated. In this way, the Chi-A protein phosphorylation lead to Chi-B phosphorylation. This Chi-B protein phosphorylation, will lead to demethylation of the MCP protein. The time to decrease by the distance or repellent. So in this way, remember, when the mission will complete, the Chi-B protein activation will occur. This Chi-B protein after the mission complete and the more attractant saturation due to the closer and closer will activate more Chi-B protein, which that will lead to demethylation of the MCP protein. So anyhow, in this way, again the mechanism is that the Chi-A protein will activate the Chi-Y protein, and this Chi-Y protein will activate the motor, flagellar motor movement, the clockwise and anti-clockwise. The time to decrease distance are repellent. So in this way, here you can see the important thing is that whenever the bacteria will close, so the Chi-Y protein will be deactivated. The dephosphorylation, the complete mobility mission will lead to dephosphorylation, and when closer to the attractant. In this way, the bacteria will find out the location, and the bacteria will no longer invest the energy. So the chemotactic and phototactic movement in bacteria can be positive or negative on the basis of situation. For example, bacteria want toward or away from the chemoattractant source. So for example, the bacteria also like and dislike. So in this way, bacteria dislike and like, the thing is that bacteria want to eat or want to repel itself. I hope you make sense about that. If you like my video, please make sure to subscribe, like and share. So see you in the next video. For this, bye. Allah Hafiz.